Hello everyone, this is Guangbin Luo. I'm a postdoctor from the University of Copenhagen, Denmark. Now I'm going to give you guys a brief introduction of our project. We are using genome editing method to accelerate the domestication of intermediate wheat grass. We are interested in 4 domestication traits of wheat grass. The first is the yield. We are going to increase the green rains, and seed size to increase the yield of wheat grass. Also, we plan to reduce the plant height and hence to increase the yield of wheat grass. The second interesting trait is harvestability. We plan to reduce seed shattering and increase free selection ability of wheat grass. The third trait is bread making quality. We plan to reduce the content of gliadins, but to increase the content of glutens in wheatgrass grain. The last trait is self-incompatibility. As we know, wheatgrass is self-incompatible, which hampers the production of genetic purity of an elite cultivar. If we can make wheatgrass self-compatible, we can solve this problem. The underlying genes controlling the above-mentioned traits are very clear in other cereal crops. For example, in common wheat, the plant height is controlled by the gene RHT1. A deletion at the data domain of RHT1 would reduce the plant height and hence increase the yield. In common wheat, knockout of GW2 and GASA7 would also increase the green size. BTR1, BTR2Q, and QSH1 are famous genes in cereal crops which control seed cythering and also free selection. Knockout of those genes could improve the harvestability of cereal crops. LAS1 is a new gene from green millet. Knockout of LAS1 would reduce seed shattering in green millet. Based on the nucleotide sequencing alignment, those genes are very conserved in wheatgrass genome, which give us a hint that we may edit those genes to accelerate the domestication of wheatgrass. A work from Dr. Jesse Poland showed that self-incompatibility of wheatgrass is controlled by S and Z loci genes. We are thinking knockout of those genes may make wheatgrass self-compatible. In genome editing, we have to design an sgRNA for each target gene. Before we transfer sgRNA into wheatgrass, we have to know this sgRNA works. Therefore, in vivo testing of sgRNAs before transformation is very necessary. We are using protoplast to in vivo test the sgRNA of wheatgrass. We isolate protoplast from young leaves of wheatgrass seedlings, and you can see each protoplast is an intact cell. After transfected with sgRNAs, if a mutation happens in the target genes of the protoplast cell, then we know this sgRNA works. Using this method, we have tested the efficiency of lost sgRNAs in vivo. Here are 2 examples of GW2 and GASA7 using the nucleotide sequences of GW2 and GASA7 from common wheat. We did a blast in the wheatgrass genome sequence, and you can see we get several hits with very high nucleotide identity from 94% to 99%. Using the nucleotide sequences from wheatgrass genome, we design sgRNAs. When we design sgRNAs, there is a restriction enzyme in the target sequence of sgRNA. If mutation happens, then the restriction enzyme cannot cut the target sequence anymore. Here is an example of GW2. You can see here, here is the target sequence from the protoplast cell, which have not been transfected with any sgRNAs. This is using negative control. You can see the target sequences have been digested by the restriction enzyme completely. When transfected with sgRNAs of GW2, you can see the target sequencing of the GW2 sgRNA can only be partial digested, but there are some sequences left which cannot be digested. Okay, this indicates that mutation happen in the cutting set of the restriction enzyme, and this sgRNA works. We did the same thing with sgRNA of GASA7, and we find sgRNAs which works well. Before we transform sgRNAs into wheat glass, we have to establish the tissue culture system of wheat glass. So you can see here from this picture, we induce embryonic callus from shoot apical meristems. You can see after we get embryonic callus, we move the embryonic callus to shooting media, and we get shoot, and then we move the shoot to lutein media, and we get lute, and finally we get regenerated plant. In addition to using shoot apical meristems as explants, we also tried immature embryos. You can see this is immature seeds, and we get and we separate the immature embryos from the seeds, and we induced embryonic callus from the immature embryo. And finally, we get regenerated plants with both shoot and the lute. So now, wheat glass is transformable. When we do the transformation of sgRNAs into wheat glass, we have two editing strategies. The first is single gene. In this strategy, we just transform sgRNA of a single target gene into wheat glass. The second strategy is the multiplex gene editing strategy. In this strategy, we transform sgRNAs of several target genes simultaneously into wheat glass. For example, we combine sgRNAs of GW2, GASA7, and LES1 in a single vector. Then we transform this vector to wheat glass. If we are lucky, we will get a mutant in which all the three genes are mutated. After transformation, we have to detect the mutation in regenerated plants. Here is an example of the mutation detection of the plant WG34, which has been transformed the sgRNAs of GW2, GASA7, and LES1. And this picture shows the restriction enzyme digestion result. You can see from this part, we can see the target sequences of the LES1 gene could not be digested, which means there is a mutation. And similarly, we get undigested binds from GASA7 and GW2. Those data indicate that all GW2, GASA7, and LES1 in this mutant WG34 have been mutated. Okay, let's go to a summary. For most domestication genes, we have designed highly efficient sgRNAs, and now wheat glass is transformable using shoot apical meristems and immature embryos as explants, and the highly efficient sgRNAs have been transformed to wheat glass, and the mutants are coming out. Next, we are going to get more mutants and do the phenotyping. Finally, I would like to thank my supervisors, Michael Palmgren from the University of Copenhagen and Zai Xiagao from Chinese Academy of Sciences. I also want to thank Bo Su, who helps a lot in genome editing, and our collaborators from Casburg Laboratory. And also, I would appreciate for all the great suggestions from our advisory board. Okay, this is my presentation. Thank you. Any questions are welcome. Thank you.